Hello. I'm sorry that we weren't able to record this presentation directly in Zoom, and so here it is, me afterwards doing it alone and recording it so that you all can have this if you missed class today, or if you weren't able to get the slideshow up, or just wish to review this. This continues our introductory material about extremophiles. We talked last time about kind of what the extremes were and what an extreme meant and what extremophiles means, what limits are and what life is. Today we're going to get a little bit more nuts and bolts. Why are there limits when it comes to where things can live? And so here's a quote by the famous science fiction author Arthur C. Clarke. He wrote, for example, the book on which 2001: A Space Odyssey is based. And he says, the limits of the possible can only be defined by going beyond them to the impossible. This reminds me a lot of my old cycling coach who used to say that about a bike racer is that if you never crashed, you weren't close enough to the line. That was fine when it was us getting the road rash and not him. So why are there limits? Why can't things live everywhere? What is it about an extreme environment that actually prevents the survival of its inhabitants? And when organisms are faced with conditions that are near the limits or stressful to them, how is that stress managed? And how do different groups of organisms differ in their resistance to stress or their ability to live in different conditions? So let's start with the first one. What is it about an extreme environment or condition that actually prevents survival? Well, that of course depends on what that extreme is and how it affects the organism. And so, for example, if you are placed in an environment that's 40 degrees below zero, you'll be affected differently than if you're exposed to an environment with 200 grays of radiation. It's also true that very often the detrimental effects on the environment are indirect. And so it may be that it isn't the acid conditions that actually kills you, but something that the acid conditions cause. That's the limiting factor instead. I'll give you an example of that in just a moment. This picture, by the way, is the Great Salt Lake. So here's the example, alkaline conditions. And so it turns out that nothing can live above a pH about 10.5. And there are a variety of reasons for that. The picture is Mono Lake. This is a so-called soda lake here in the US, described in amusing detail by Samuel Clemens in Roughing It. You should get a copy and read that if you get a chance. One of the issues with alkaline environments that causes trouble is saponification of membrane fatty acids. So alkaline material, for example, Drano, if you touch this stuff with your hands, it feels slippery, it feels soapy. That's not because the Drano itself is slippery or soapy. It's because as soon as that Drano touches your fingers, it saponifies the fat in your skin. Saponification is the process by which the alkaline hydrolyzes the ester backbone of membrane lipids, separating the fatty acids from the glycerol phosphate. And so the alkaline actually generates soap on your skin as you rub your fingers together. This is how people used to make soap. In fact, if you buy natural soap, this is how they still make it. You take animal fat and you mix it with something really alkaline, typically ashes from a fire. Ashes are highly alkaline because the organic material contains a lot of metal cations like calcium, magnesium, sodium, potassium, but organic material is the acid part. the anion, like acetate and glutamate, etc. The organic acids are volatilized and burn up, leaving the alkaline salts, so sodium hydroxide or potassium hydroxide. And so ashes are alkaline, and you can mix that with animal fat and you make soap. If you leave the glycerol in, then you have glycerin soap. If you remove the glycerin, then you have regular soap. Now you can imagine that hydrolyzing the membrane lipids to make soap and glycerol phosphate is not good for membrane lipids. It kills cells. It disrupts their integrity because the membrane lipid falls apart. Alkaline conditions are also harmful for organisms because of the inability of organisms to make energy. Most organisms make energy. They make ATP by utilizing the proton gradient. And so they use oxidative phosphorylation or some other mechanism through electron transport to make a proton gradient. It's a little bit like a battery. It actually is a capacitor. It's a kind of a battery. With high protons or high positive charge on the outside and a negative charge and low protons on the inside. And by leaking those protons in slowly through an enzyme, they can utilize the energy of that gradient to make ATP. But when you pump protons out to make the gradient, if you pump it out into an alkaline environment, as soon as that proton is outside the cell, it reacts with a hydroxide to make water, and you don't have any protons outside. And so you don't have any protons on the outside to leak back in to make ATP. And the result is that energy metabolism fails at high pHs. Another thing that organisms deal with is membrane proteins are destabilized. And so membrane proteins have particular structures, and part of that structure relies on the presence of hydrogen groups forming hydrogen bonds and other sorts of interactions on the surface of the protein. And in a strong alkaline condition, some of those proteins aren't attached strong enough to stay on, and they're deprotonated. And the result is the change in charge and electronegativity of the surface protein, and often those proteins become denatured or don't function properly. One of the indirect effects of alkaline conditions is the presence of toxic metal ions. These metal ions, things like selenium and arsenic, are present in low concentration in the rocks and sediments around them, but they're found in insoluble forms under most conditions. But under alkaline conditions, the alkaline leaches that, extracts it from the rock, and brings it into solution. And as a result, Mono Lake, for example, the water is toxic to drink, not because of the pH, but because it's full of arsenic. And the other thing is that often alkaline environments are also saline. So we call this a soda lake. Yeah, there's a lot of alkali in the water, but there's also a lot of salt. Sodium chloride, potassium chloride, as well as carbonates and other stuff like that. And so you have to deal not only with the extreme pH, but also the extreme salinity. Intrinsic limits also exist, and it's interesting to think about why things can't get, for example, bigger or smaller. There are no insects as big as a person. Why is that? Well, probably the reason for that is twofold. One of them is has to do with how strength works, and the fact is that an insect our size, the legs wouldn't be strong enough to hold up their mass. Secondly, insects have an open circulatory system, which is an efficient way to deal with circulation in a small creature, but it doesn't work very well for large creatures. Here's an example of a small organism. This organism is Pelagibacter ubiqui. It's one of the most common organisms in the world. Very, very small, less than half a micron across, too small to be seen with a typical microscope. Really abundant in ocean water. The issue with it getting smaller, and this is probably the limit for how small an organism can get. So if you look at this bacterial cell, you'll see that it looks like it has a very thick envelope compared to other pictures of bacteria that you may see. And the reason for this is bacterial envelopes are the same thickness regardless of how big the cell is. It's two membrane bilayers and a thin peptidoglycan wall between, and that membrane doesn't get any thicker or thinner depending on how big the cell is. And so when you get to small cells, the relative thickness of that envelope is greater and greater. And in this particular case, although it doesn't look like it in this picture, the cell envelope comprises about 20% of the cell mass. It also has a problem with DNA. In a bacterial cell, you have to have a certain set of genes. This organism lives in the environment and makes a free-living organism. It makes a living like any other ocean creature, and it has to have all the genes to do that. And genes aren't bigger or smaller based on the size of the cell. And so in this organism, just the plain DNA in the nucleoid of this cell makes up 30% of the dry weight of the organism. If the organism got any smaller, it would have to throw away some of those genes because that's about the only place where it's got any mass it can get rid of. An issue with getting smaller, one of the primary issues, is that the volume to surface ratio decreases. The smaller you are, the more surface area you have relative to the amount of mass you have. That's an advantage for this organism, Pelagibacter. What that means is any square millimeter of surface has to serve to acquire the nutrients for a smaller volume of cytoplasm. Now, why does the surface to volume ratio change as you change in size? It's because the volume of the organism increases with the cube of the size. If you double the length of an organism, you have 8 times the volume, 8 times the mass, but only 4 times the surface area, because surface area changes with the square. That's what square and cube mean, right? You're talking about 2-dimensional or 3-dimensional objects. So in an organism like Pelagibacter, that's why it's so small, because that means it has much more surface area relative to the amount of cytoplasm it has to feed. This can be an advantage for large organisms. So for example, large organisms have a large mass generating probably a certain amount of heat. And if you've got a small surface area, it's the surface area from which the heat radiates. And so if you live in a cold environment, being large is an advantage because it means that it's easier, you use less energy to generate and retain heat. So if you think about Arctic environments, you think the animals that live there tend to be large. Whereas in tropical environments, the opposite of the issue is that you may generate more heat than you're able to get rid of. And so tropical organisms tend to be smaller, and they have to have ways to blow heat off more efficiently. Small size also is a problem for predation. The smaller you are, the more likely you are to have predators. Whereas the opposite, of course, if you're big, then you have less problem with predators. This is how elephants do it. And so each different kind of extreme has different kinds of limitations. Acid also has an issue with membrane lipids, although it's a bit different. Also has an issue with protonation of proteins on the surface. An interesting aspect of the proton gradient with acids is that you've already got a proton gradient, even if you don't do any work. That is, there's lots of protons on the outside. That's what acid means. The problem is that you still have to pump protons out because if you leak protons in to make energy, you're acidifying the cytoplasm. And in order to maintain the pH of the cytoplasm, for every proton you pump in, you have to be able to pump one out again. And here the problem is that you're pumping against such a large gradient already that some organisms are unable to push their electron transport system against such a strong gradient. High temperature, low temperature, drying, irradiation, large size, salinity, oxidation. These are all limits. Each of these has different things that are harmful to the organisms, different things they have to deal with. And we'll talk about what those are as the semester progresses. But let's talk about one of them right now. There are some specific adaptations that organisms have. And so here's an example of heat. One of the primary things that limits your ability to survive in heat is the fact that membranes, the lipids in the membranes, are tuned to work best at a specific temperature. Membranes are two-dimensional fluids. The lipids and the proteins in a membrane float around in that membrane. They have to stay in the membrane, but in that two-dimensional field, they can move around. And this is important to the function of a membrane, any membrane. If you decrease the temperature, those membrane lipids solidify. It goes below their solidification point. Just like if you have lard and you cool it down, it solidifies. So too, the lipids in a membrane will solidify, and things can no longer diffuse in the membrane, and the membrane function fails. On the other hand, if you heat it up too much, then things in the membrane are too mobile. And in fact, you start to lose lipids and proteins from the membrane itself, and the integrity of the membrane fails. And this is modulated in organisms by changing the composition of the lipids. And so if you grow an organism at one temperature and then shift it to a different temperature, over a fairly brief period of time, it will turn over those membrane lipids to membrane lipids that are more appropriate for the temperature it's now growing at. Another thing is DNA supercoiling. If you think about DNA, it's a double helix and it's all hydrogen bonded up. And it turns out that the melting point of DNA under physiological conditions is about 65 degrees centigrade. And that's a good thing. You don't want the strands of your DNA coming apart all the time. But you do need to be able to separate the strands of DNA to do the kind of things that DNA is needed for. To replicate it, you have to separate the strands, or to transcribe it into RNA. The RNA polymerase has to be able to separate the strands to copy it into RNA. And if you're too far away from the melting point of DNA, then these enzymes aren't physically capable of pulling those strands apart. And so organisms use things like histones and supercoiling. Supercoiling is the torsion on the helix. And so it's just like taking a hold of the helix and cranking it to separate the strands, cranking it open. And so they use these mechanisms to tune the DNA so that it stays together, but it's close enough to coming apart that the enzymes can open them up for what they need to do. RNAs in the cell, things like the ribosomal RNA, etc., are tuned, and we're going to talk about that in the next slide. Proteins also have to adapt. Proteins, think of an enzyme. An enzyme can't be too rigid, because if it's too rigid, then substrates can't get in and products can't get out. It has to be flexible, but it can't be too flexible, because if it's too flexible, it will flop around and then come apart. Think of an enzyme as being made of jello. It's jello-y enough that stuff can get in and out, but it's not so jello that it just comes apart. And so if a protein in an organism that's adapting to a lower temperature has to increase the interactions inside that hold it together and make it more rigid, they have to loosen it so that it still is fluid, still is jello-y at the lower temperature. It doesn't get too rigid. And if you grow the organism at a higher temperature, then it's got to make that protein more rigid, or increase the things holding it together so that it's at the right rigidity at that higher temperature. Small molecules are actually quite a difficult problem. And so if you think about all of the vitamins and cofactors and things like ATP and GTP in the cell, most of these things really don't have a short half-life at elevated temperatures. That's why in the lab we have to store them in the freezer. And so most organisms don't keep these giant stockpiles of these things around, because if you have a lot of it around, then you're always losing it to degradation, to denaturation, or to decomposition. And so what organisms do instead, particularly organisms that live at high temperatures, is they make it and use it immediately so that there isn't time for the molecule to decompose. Well, let's talk about RNAs in particular. And so here are two RNAs. These are homologous RNAs, so the corresponding RNA in two organisms. The one on the left is Thermatoga maritima. This is a bacterium that grows at 85 degrees centigrade. On the right is the same RNA from Synechococcus PCC 6301. This is a mesophilic cyanobacterium. It grows optimally at about 15 to 20 degrees centigrade. Now these two RNAs, you can see they're quite similar, and they're all made up of a single RNA strand that's all folded up. And it's folded up using Watson-Crick base pairs. Watson-Crick pairs in RNA are GC, of course, and AU. It also turns out in RNA that GU pairs are perfectly reasonable. They're energetically nearly as good as AU pairs. But if you look at these RNAs a little more closely, what you see is that if you scan around, you'll see that the Synechococcus RNA, the double-stranded regions have a lot of irregularities in them. Mismatches, things like GG pair or a bulged C sticking out, or loss of GU pairs. You might think of these things as flaws in the RNA structure, particularly when you look at the RNA of Thermatoga that doesn't have these things. And in a way they are flaws, but they're purposeful flaws. These are irregularities in the RNA that increase the flexibility of its structure. And it needs that flexibility at low temperatures in order to bind its substrate, carry out the chemistry it does, and throw the products away. However, that added flexibility in that RNA means that if you heat it up to 85 degrees where that other RNA has to work, there aren't enough good hydrogen bonds holding it together to keep it integral. And what happens is the RNA unfolds in solution. Whereas the RNA on the left, the Thermatoga one, doesn't really have a lot of irregularities in it. It's a nice tight structure, which makes it very solid, which means it can work really well at high temperatures. But at low temperatures, it lacks flexibility. And as a result, it's fairly inert. It doesn't work very well at low temperatures because when the substrate comes in to bind it, it's not flexible enough to allow that accommodation as the thing slots into place. These things are tuned. And in fact, if you look at this, with even what I've just told you, you ought to be able to take two homologous RNAs from two different organisms, and if one's a thermophile and one's a mesophile, you ought to be able to tell them apart. Certainly an expert can do this at a glance. The interesting thing is that nobody can do this for homologous proteins that live at different structures, at different temperatures. We don't understand enough about protein structure and adaptation to be able to look at a protein three-dimensional structure and tell you which one, whether it grows at a high temperature or at a low temperature. So how are stresses managed by organisms? A lot of the stress response that organisms have are generic. That is, the organism is under stress and it tries to manage that stress the same regardless of what the stress is. And so the primary mechanism organisms use is something that's called the heat shock response. It's called the heat shock response because it was discovered as a response to increases in temperature, sudden increases in temperature, and it was first studied after you take organisms and shift them up 5 degrees in temperature. It turns out that the same response occurs for other kinds of stressors, particularly things like acid or alcohol exposure, etc. What happens in the heat shock response is that organisms suddenly shut off the synthesis of most proteins. But it turns way up the synthesis of a handful of the so-called heat shock response proteins. What these proteins do in general is shut off DNA replication and deal with the fact that that increase in temperature has had a bad effect on a lot of the cell's proteins, typically unfolding them. And the organisms then have to deal with the fact that all of a sudden they're full of unfolded proteins. Even newly made proteins they've just synthesized get unfolded. And so a couple of the primary proteins in the heat shock response are chaperones, proteins that facilitate the refolding of denatured proteins. And once those proteins are refolded, once the organism has recovered from that initial shock, then the heat shock proteins are turned back down and general protein synthesis can occur once again. And one other response found primarily in bacteria is the so-called SOS response. This is a response to cells that recognize genetically that something is seriously wrong with its DNA or DNA replication. And so, for example, the SOS response can be induced by bacterial phage infection, the infection of that bacterium by a virus. Interestingly, the SOS response can also be responsible for an inactive virus in a cell to become active, because if a cell is stressed out, it may be in serious danger, and it's in the evolutionary interest of the virus to bail out, like rats fleeing a sunken ship. One of the best understood stress responses is the so-called stringent response in bacteria, which is a bacteria's response to starvation. What happens in starvation in bacteria is that this first shows up as a signal when protein synthesis fails. The amino acids it needs to make proteins run out. And so what happens then is that the ribosome, as it trundles along a messenger RNA, the ribosome stalls because it doesn't get the amino acid it needs to tack onto the end of the protein. Stalled ribosomes send out a signal, and that signal does a number of things. It shuts down DNA synthesis, it shuts down further protein synthesis, it shuts down the synthesis of more ribosomes. And the ribosomes are typically the rate-limiting step in bacterial growth. And if your ribosomes aren't getting the amino acids they need to make proteins, there's no reason to be making more ribosomes. And that regulation is called the stringent response. You'll learn about these in genetics and in biochemistry as you move through your career. Well, it's pretty obvious that different groups of organisms have different levels of resistance. When you think about extremophiles, probably you think about archaea. And the best known extremophiles are archaea. And although there are a lot of archaea that live in mundane environments, it certainly is true that most of the archaea we know about are extremophiles in one way or another. But bacteria are everywhere. They're abundant, they're very diverse, and just by the fact that they are so diverse and so abundant and so everywhere, there are also a lot of extremophilic bacteria. Eukaryotes, on the other hand, are not generally as extremophilic. There are some extremophilic fungi, and there are extremophiles here and there. But the reality is that the common ancestor of eukaryotes was not an extremophile. And eukaryotes are so complicated in their biochemistry and structure that they have found the adaptation of that complexity to life in extreme environments generally pretty difficult. It is also true that tolerance to extremes are often coupled. And so I mentioned Lake Mono that is both alkaline and high salt. And it turns out that the resistance of an organism to salt and acid and base are often linked. It's often found that organisms that can live at high temperatures and high acid are one and the same. A great example is organisms that are resistant to high levels of radiation also tend to be resistant to drying. And this is because the damage done by radiation is typically DNA double-strand breaks. And the resistance to drying, or the thing that drying damages you, is also by making double-stranded DNA breaks. And so the damage is the same in both of those cases. And so if you're resistant to one, you might be resistant to the other. And again, it's often true that extremes are often opportunities, not just difficulties. And so, for example, if you're able to adapt to life at high salt in, for example, a salt lake, you're also likely to find that there aren't very many predators against you. And so that's if you can adapt to high salt, you can probably use that as a way to avoid predation. Likewise, if you with temperature or acidity. Growing at a really high temperature might be difficult, but it's also true that the equilibrium chemistry is different at high temperatures. And at high temperatures, there are chemical reactions that you can use to extract energy that aren't available to organisms at low temperatures. And so that's an opportunity you might be able to take advantage of as well. So don't think of extremes just as hazards or as difficult things, but they're also opportunities. All right, so next week we're going to go with a specific example, both in terms of extremes. We'll be talking about high temperatures and acidity, and also specific in terms of a particular place, Yellowstone National Park. Yellowstone is the highest concentration of hot springs in the world, and we're going to talk about a variety of hot springs and so forth, and the organisms that live in them. It might look a little bit like vacation slides, but instead of me showing you a bunch of pictures of beautiful mountains and moose and bear and bison, you're going to see one picture after the other of slime living in hot springs of the park. And we'll talk about what's in those organisms, how they live, what kind of conditions they deal with, etc. And next week will be the first discussion session where you're expected to respond to prompts and so forth. But we'll talk about that next week. For now, I hope you have a good weekend, and please stay safe. I'll send you a Zoom invitation for next week's thing, the middle of next week. Thanks so much. Take care. Bye now.